Hi everyone, I'm Shuze Wang. I'm a graduate student at University of Michigan. Today I'm going to talk about single-cell chromatin accessibility, delineates cellular identities of the neonatal organ of Corti. Hearing is mediated by the organ of Corti within the cochlea of the inner ear. The organ of Corti is composed of a variety of cell types including hair cells and supporting cells. Transcription factor TF binds to the DNA regulatory elements to control gene expression in a spatial-temporal manner, therefore enabling a single genome to give rise to diverse cell types. However, gene regulation mechanisms during cochlear development and differentiation are not fully understood. As if for transposees accessible chromatin sequencing, ataxic profiles accessible regions of chromatin indicating active regulatory regions, which enables us to better understand comprehensive epigenomic landscape. Our aim is to identify transcription factor regulating differentiation and maturation of the organ of Corti. For the experiment, we dissected cochlear ducts into apical and basal compartments. Then we used flow sorting to isolate individual cells and specialized hair cells and supporting cells. Libraries of sorted cells were prepared using the 10x single-cell ataxic and single-cell RNA-seq protocols. To analyze the data, we clustered to determine their identities. Next, we conducted functional analysis, such as TF classification, gene regulatory network, 1D spatial reconstruction, and development trajectory analysis. Starting the downstream analysis, the first step is cell type identification. For single-cell RNA-seq, we ran differential analysis and we identified differentially expressed genes for each cluster to determine cell identities. Here we show top 100 DE genes sorted by p-value for each cluster. Similarly, we identified differentially accessible regions for our 6 ataxic clusters. Here we show top 100 DARs for each cluster. To identify the cell types for single-cell ataxic dataset, we calculated a J-card similarity matrix, which compares the similarity between DE genes from RNA-seq and annotated DARs from ataxic. The rather the more similarity between two datasets. For example, cluster 1 in ataxic has the most similarity with hair cell cluster from RNA-seq dataset. Next, we checked the performance of individual genes at single gene level. We picked one gene per cluster for visualization. The 6 clusters are distributed along the y-axis and candidate genes are plotted along the x-axis, each divided into a chromatin accessibility column and a matching violin plot for the transcript level. For example, the POL4F3 locus was determined to be differentially accessible in hair cells and POL4F3 transcript was found to be differentially expressed in hair cells as well. Has5 is a marker for the supporting cell group and so on and so forth. To achieve our goal, we inferred TF activity in maturing hair cells and supporting cells. We assumed increasing the expression level of an activator will increase the average accessibility, and increasing the expression level of a repressor will decrease the average accessibility. Based on the assumption, we calculated the correlation between the significance of mRNA expression level and the chromatin accessibility from t-test for both hair cells and supporting cells. To validate our classification, we featured 4 TFs, including 2 activators and 2 repressors, in mRNA expression level, chromatin accessibility, and footprints. Then we want to identify downstream target genes of TFs. We were inspired by Cynic algorithm. However, the major novelties of our analysis are we integrated single-cell ataxic accessible regions and we identified activators regulons and repressor regulons. To be more specific, we first identified co-expression modules. Secondly, by integrating single-cell ataxic information, we identified putative TF binding sites from accessible regions where the TF binding sites is within 50 kb of the TSS. Based on that, we identified regulons, which are groups of genes that are regulated as units. Then we calculated the activities of the regulons by considering TF expression for each cell. We applied our gene regulatory network analysis to hair cells and supporting cells. We generated regulon activity heat map with hierarchical clustering at single-cell resolution. We observed that 801 LHX3 regulons are highly activated in hair cells, while SOX2, SOX10, GLIF1 regulons are enriched in supporting cells. Previous publications showed that developmental of the organ of Corti precedes ingredient. Focusing on hair cell cluster, we identified differentially expressed genes and differentially accessible regions between apex and base. Then we used those features to generate 1D PCA plot resolving individual cells' relative position along the tonotopic axis. Next, we featured a candidate gene, PKHD1L1. PKHD1L1 is highly expressed and more accessible in the basal compartment compared with apical compartments in both ataxic and RNA-seq. In summary, we predicted the activating and repressing function of developmental TFs in cochlear hair cells and supporting cells. We inferred TF associated with downstream target genes promoting the maturation of hair cells and supporting cells. We resolved the original structure of organ of Corti along the apex to base axis, and we mirrored the gradual changes in the epigenomic landscape during development along the tonotopic axis of hair cells. And thank you everyone for listening.